When cells in our body sense a lack of oxygen, they upregulate the production of red blood cells. This increase in red blood cell production is mediated by an important molecule called the hypoxia-inducible factor. For his discovery of this really key molecule, Gregg Semenza won the Lasker Award for Basic Medical Research in 2016. And this molecule, as you can imagine, touches every facet of life. Because as my guest today puts it, without oxygen for 5 minutes, you end up dead. Hi everyone, and welcome to Doc Talk. I have the pleasure of having with me today the 2016 Lasker Laureate, Professor of Medicine, Pediatrics, and Oncology at the Johns Hopkins Institute, Gregg Semenza. Dr. Semenza, welcome to the show.
Thank you.
Sir, in reading about your work, how you discovered the hypoxia-inducible factor, there were a lot of things that stood out. And we hear about serendipity in science all the time. But to me, your story of how you discovered this factor was a remarkably perfect example of this. Because if I remember correctly, you contacted a biotech company in Massachusetts because you wanted to acquire certain products to study hemophilia, another blood disorder. And they said, well, we have clone for this EPO gene, which turns out has a role in how cells respond to oxygen. And they shipped it to you without you even asking for it. So how do those events come about? And how do you interpret them in the larger context of your work?
Yeah, well, I think that the serendipity is one of the really things that makes science exciting, because you never know really where things are going to lead. And oftentimes when you have a hypothesis that turns out to be incorrect and you find out something you weren't expecting to find out, that leads you off in an entirely new direction and to a whole series of new findings that were not at all expected and turn out to be very important. So it's one of the exciting parts of research, that things don't occur in a straight line, and that you can follow things wherever they lead, and that often leads to exciting discoveries.
Now, I also read that what really got you started in this field of medical genetics was when a family friend of yours had a child with Down syndrome. So what was it about these genetic disorders that really captivated you?
The concept that someone has a disease that they've inherited, for which there's no aspect of their lifestyle that's responsible, that, it's basically just a twist of fate, that they've inherited a gene or a chromosome that results in a oftentimes profound disorder that affects many aspects of both their physical and mental well-being. To me, that was very in a way. I guess I found that most compelling in terms of thinking about medical care. It was remarkable the way these families could adapt to these really significant demands that were being put on them by the child's, or in some cases multiple children's, disease, and really showed you the strength of character that people have to be able to really maintain a normal lifestyle despite having great burdens placed upon them by a family member's disease.
That's really profound and incredible. And along those lines, sir, one of the concepts we learn in science and medicine is that a lot of environmental triggers interact with the genetics that a person has. And speaking of this brings me to a topic which is quite popular in the media these days, which is of faulty reporting, right? So it seems like, for many decades, there was almost everything that was shown to be both positively as well as negatively associated with smoking. And for a long time, that gave, say, for instance, the tobacco companies a reason to say that, you should continue to smoke. There may be some faulty science behind what is being presented. So as people are grappling more and more with this concept of sometimes picky and selective reporting of scientific facts. What would you advise the lay audience or the common man to sort of really think about as they are reading about science?
Yeah, and I think that it really is important to understand that you do have these environmental stresses and you have predispositions, and it's the interaction between those that often results in disease. And what that means is that someone may be lucky enough that they can smoke 30 packs of cigarettes, a year and not get cancer, and somebody else will just have a very light smoking history and will end up with lung cancer, right? Or even in the worst case, somebody who is subjected to secondhand smoke and will develop lung cancer, right? And part of this is understanding that there are no absolutes. That there's no 100% association, and that so people who say, well, who can point to someone and say, well, this person didn't get that disease, therefore, the concept can't be correct. That's just a lack of understanding of the fact that all of these are sort of they increase the risk. It doesn't make anything necessarily absolute, but that doesn't mean that there's no relationship between the two just because they're not associated 100% of the time.
So there's no doubt that some of the biggest and most impactful discoveries made in science have come from study of open-ended questions, which I think is in contrast to industry university partnerships or projects that are largely driven by those goals. So as you've moved along in your career, have you preferred one particular type of questions versus the other, and how has that changed?
Yeah, I guess my own approach to science is sort of in part based on my training, because I have both an MD and a PhD, and so I've kind of viewed my role as bridging the basic science and the clinical medicine. And so we've been particularly interested in trying to follow questions from the very molecular side of it all the way to the clinical, and hopefully in some cases to developing new treatments for diseases. And so it's the ability to kind of connect the dots across the scale from the very molecular mechanisms to the outcome in terms of patient mortality or treatment. That's, to me, most fulfilling to be able to take that approach to science.
Do you think physicians who have a really firm understanding of basic science end up becoming better doctors when they see patients?
Yeah, I do. We have a lot of students in my lab who are on a pre-med track at Hopkins, and they're interested in going to medical school. And what I tell them is that the approach to science that they'll learn in the lab is also a useful approach to patients, because the idea is you take a systematic approach to trying to solve a problem that allows you to get to the answer most effectively and not end up going down some blind alley that will take lots of time. And in the case of research, it may a lot of expense. In the case of a patient, of course, it may result in the patient's loss of life. So that and the other is the attention to detail that is necessary for very fine molecular experiments to work. That same attention to detail is obviously very important when you're treating a patient, because if you give a patient 10 times more chemotherapy than they're supposed to get, of course there'll be very adverse consequences to that. And unlike the science, where you can do the experiment again, that may not be the case in the patients. And so I think the skills are transferable, and that having training in one will have benefits in the other.
Very wisely put indeed, sir. The other thing I wanted to ask you is that, say, for someone like you, you went to Harvard for undergrad, you were at Penn for your MD, PhD, and went on to Duke for your residency, and you're now at Johns Hopkins as a professor. So there's, a chain of institutions that are regarded as the mecca in biomedical research and in higher education. How much does pedigree really matter? And how does it change what you are able to do and accomplish if you go to one of these top 5 or top 10 places versus if you don't?
Right. Well, I think, first of all, from a practical point of view, that success is based on taking advantage of opportunities. And that at many institutions, there are more opportunities that could possibly be taken advantage of than any one person could utilize. And there's a joke about a guy who goes to a college tour, and they go to the library, and the tour guide says, we have 2 million volumes in our collection, and the student says, but at Harvard they have 20 million books in their collection, and the guide says, well, when you finish reading these, we'll get you some more. So that's the idea, is that probably in most situations there are opportunities to be taken advantage of. Yes, there are certain reasons why certain institutions are highly regarded, and those, are substantial reasons. But that does not mean that that's the only path to success by any means.
Mm-hmm. Thousands of people who enter a scientific lab at some point and then realize this is too hard, or this is not working out, or I'm not getting the results that I want. How were you able to overcome that barrier, and what would you tell them?
Yeah, well, actually, it was worse than that, because I started in one lab and started on a project, and that project didn't seem to go anywhere. And so I sort of switched to a different project within that lab, and that project also was unsuccessful. And so and that was after 1 year. Then I decided to switch into an entirely new lab, and study this the family with beta thalassemia and look for a mutation in the beta globin gene. And after 1 year of doing the analysis, we found out that the result we got was due to contamination and was completely invalid. So we had to start over again. So at that point, yes, I was very depressed, and there were a set of twins in my med school class, and their father was a psychiatrist. So I went to see him, and I told him what had happened. And I told him I was very depressed as a result. And he said to me, well, it sounds like you have good reason to be depressed. And so then I didn't feel so bad. And I went back to the lab, started over again, and this time, things worked out. And actually, my first mentor when I was an undergraduate and worked in a lab at Children's Hospital in Boston, he used to say search and research. And so there's a certain amount of perseverance that's required for success in science. One has to view a failure as not some cause for depression, but more as a challenge, an obstacle that needs to be overcome. And so if you don't have that sort of enthusiasm for overcoming challenges and obstacles, scientific research may not be your cup of tea.
Yeah, I totally agree. And sir, the other thing that's incredible about you, needless to say, is that you are a member of all these wonderful organizations: the National Academy of Sciences, the Institute of Medicine, the American Association of Physicians. And there's no doubt that it is an honor to be elected as a member in these organizations. But to you, what does it really mean to be a member of all these organizations, and how does it actually change your life?
It's nice to have the validation of these groups, which are made up of lots of distinguished scientists and physicians, that you've made a certain contribution to biomedical research that is sort of worth noting, and to sort of gain election into a group of people who have made very substantial contributions to biomedical research obviously is a real honor. And also from a personal point of view, of course, to get to meet people who have all these accomplishments is also is a wonderful opportunity, and to get to work with them on different projects, like on the editorial board of PNAS. Which is a lot of work, but it's also a lot of fun, and is the way that I contribute to the National Academy of Sciences, so that I'm I'm making some contribution to the organization rather than just reaping the benefits of having this on my CV.
Is there a burden to sharp brains?
The people who are successful in science, they may not be terribly different than the people who are less successful in science, or at least less recognized. But it may be, to a certain extent, a series of fortunate occurrences, and maybe the particular area that they're working in that's of more general interest, and therefore receives more attention. And particularly for students to understand, no, there's no reason why you can't get to that point as well, because this guy started out where you are, no different.
I imagine as a PI, when a new grad student comes to you, or when you hire a postdoc or other staff, many times these people don't realize what their strengths are, and it's up to a mentor to actually figure out that this particular person who I've hired is really strong in field A, and therefore I should give him a project that fits him well. So how do you really scout that?
Well, part of that is I ask people when they come into the lab, what is their goal? So what kind of a career is this position going to train you for? And then I explain to them that whatever career path they choose, I will support them, but that I will also explain to them what's required to get to their goal. Because some people may have aspirations that are here and motivations that are here. And I explain that either this has to come down or this has to come up so that they'll meet, so that you'll be successful. And so depending on what the person's goals are, I'll suggest a different course. So someone who's interested in an academic position, well, they should at some point write a grant, because that's such a big part of it. Someone who may be interested in going into a industry position, that would be less important, right? And really, wouldn't necessarily be a good use of time.
Right.
So I think that's a big part of it, is that people, understand what's required to reach, whatever goal they set. And then, of course, I'll do whatever I can to help people reach those goals. And to me, that's the most exciting thing. So when my students win an award, I'm much happier than if I win an award. It's really a great moment to see a student flourish like that and get recognized. Yeah, I love it.
That's really incredible. Sir, in a career that has spanned so many years, and some marvelous discoveries that have really impacted science in an indescribable way, what was your aha moment in your career, and what lies next for you?
Well, obviously, when we discovered HIF1, that was sort of the big moment. And of course, at that time, we were looking at a very focused question. We simply wanted to know, how is it that the human erythropoietin EPO gene gets turned on in response to low oxygen? And and we found this factor. We showed that it bound to the gene and turned it on. But then as we went on, we discovered that it was regulating the expression of other genes and responsible for other responses to hypoxia, like making blood vessels within ischemic tissue and regulating the metabolism of cells so that they can continue to make energy regardless of what the oxygen concentration is. And the way that it's just kind of expanded to include thousands of genes and many different common causes of disease. So cardiovascular disease, cancer, and to have the opportunity potentially to impact on those diseases by developing new therapies. So the reason that EPO was originally cloned was that because individuals with kidney failure did not make EPO, because the kidneys are the source of EPO, and they would require transfusions, and then they would be at risk for transfusion-associated viral diseases. So with the development of recombinant EPO, the protein could be injected. These individuals would make red blood cells because there's nothing wrong with their bone marrow, and would not require transfusions. But this still required injections of a recombinant protein every month. And now, with the discovery of HIF1 and the proteins that regulate it, it's been possible to develop drugs that induce HIF1 activity, and these can be taken by mouth. And so it may be possible that they will replace the injections of recombinant EPO in people who have renal failure and are anemic. So that's, I think, will be the first sort of application of our findings to treatment of disease. There's also, on the other side, HIF1 does things that actually promote cancer, and so there we'd like to block its activity. And there's a drug that blocks a related protein, HIF2, that's now in clinical trials for renal cancer. So it'll be interesting to see whether that turns out to be an effective drug as well. But that's, again, the end goal and the most satisfying part of making basic science discoveries is actually seeing them translated to new treatments for patient care.
It's really fascinating. Sir, next, I want to ask you a set of questions for which you have to respond with the first thought that comes to your mind. This is a rapid fire round. So are you ready?
Okay, I'm ready.
One quality that separates a good doctor from an exceptional one.
Meticulousness.
One quality that separates a good scientist from an exceptional one.
Meticulousness.
If you were a medical student today and were doing this all over again, what's something you would have done that you did not do 30 years ago?
I guess taken more clinical rotations in more different specialty areas.
A scientific concept or a paper whose understanding really changed the way you looked at science?
I guess my inspiration was the work on the lac operon by Francois Jacob and Jacques Monod, which really was the sort of first understanding of how genes can be regulated at the level of transcription. That was sort of my inspiration for what we eventually learned in terms of the way that HIF1 can regulate the transcription of many human disease in response to an environmental stimulus, much like the lac operon is in bacteria.
One life skill that science has taught you without you even realizing it over the years?
Well, I think, you start out in science and you think everything is based on fact and is a meritocracy, and at some point you realize that it's a profession that's based on interpersonal relationships, and that's an important part of the scientific process, is sort of interacting with your peers and moving science forward in a way that's rather personal. And is another part of the sort of fulfilling aspect of the career, because you get to make friends with people all over the world who share a common passion for science.
Well, thank you so much once again for being here today. Thank you all for watching. I hope you enjoyed this segment. And until next time, this was Divyansh Agarwal signing off on Doc Talk. Thank you so much, Dr. Semenza.
You're welcome.
Thank you.